Good morning, children. This is my eighth video on the chapter Human Reproduction, and today's topic to be explained is embryogenesis. In the previous video, I had explained the physico-chemical events that take place during fertilization in humans. So I had explained the place of fertilization, the various events in six stepwise orders. The first was insemination or ejaculation. Second was the movement of sperm. The third was arrival of ovum. The fourth was capacitation or activation of sperms. The fifth was fusion of gametes. And the sixth and the last was amphimixis or karyogamy. After this, I also explained briefly about the determination of the gender or the sex of the baby. So today's topic to discuss is embryogenesis. The term is made up of two parts, embryo and genesis. So this means this is the development of embryo from a fertilized egg into its subsequent development into a full-fledged organism. Like just see, this is a fertilized egg or a zygote, which is the product of fertilization, right? That is fusion of male and female gamete. So this fertilized egg will change into an embryo and finally into a full-fledged organism or a baby. So this entire journey of changes from a fertilized egg through the stage of embryo till we complete the formation of a complete organism is called embryogenesis. There are 5 steps under which this takes place. First is cleavage or segmentation. The second is blastocyst formation. The third is implantation. Fourth is gastrulation. And fifth is organogenesis. So in your course, children, gastrulation is not mentioned, so you don't have to do, but the rest of them you have to study. All right. So let us begin with the first one, that is cleavage. The term cleavage also can be said as segmentation. And shortly, it means, in a very simple meaning, this is repeated mitotic divisions in the zygote. That means the same zygote which is formed or the fertilized egg will undergo repeated mitotic divisions till it transforms a single fertilized egg cell into a hollow spherical and multicellular blastula. This is very interesting and very simple. Look at this. Here is, I'll show you more clearly, it's here. This is a zygote or a fertilized egg. Now in humans, within 24 hours after fertilization, the first division or cleavage takes place and the cell divides into two blastomeres, right? So here, this is the two-cell stage. And where exactly is this structure just now at two-cell stage? It is in the upper portion of the fallopian tube or the oviduct. All right. Now, within 40 hours, now the second division is within 40 hours after fertilization. This is called as the second cleavage. And here, the division, it further occurs. This divides it into 4 cells, right? That means from one cell, it became two-celled, that is blastomeres, and now finally into 4-cell stage, right? So this was within 40 hours after fertilization. Now, after this, the third cleavage or the division occurs, and this is 3 days after the fertilization. So here, structure is now 8 cells. So it is becoming double, 1 into 2, then into 4 and 8. And it will not stop here. This will continue with a subsequent number of cleavage divisions, one after the other, in a very, very orderly manner, right? Till the structure now from 8, see here it was 8 cell, now it becomes double, that is 16 cell structure, and we call it as morula, right? So during this period, that is at this situation, the embryo now slowly moves down the fallopian tube towards the uterus, right? And here the structure is 16 cell structure and we call it morula. So what is morula? It is a solid ball of 16 to 32 blastomeres. Can you see here? This is still surrounded by the zona pellucida, the same transparent layer. I'll have to go back to the previous diagram. So here I had explained this was the structure of ovum, the zona pellucida, which was transparent non-cellular layer around the ovum or an ootid. So the same zona pellucida is still persisting around the morula, right? And this is about the same size as zygote. Now notice one thing, children, that the zygote number of cells, they are increasing by division, but the size is exactly the same, right? This actually leads to, right, a situation where we can say compactation is occurring. That is, number of cells are increasing but without the growth. And this makes blastomeres progressively smaller, smaller and smaller. We call a term as compactation, right? So now what happens further? Till here we have understood this is 16 cell stage, the morula, or this could be 16 to 32 celled, and zona pellucida still there, right? Now here you can see there are a group of large centrally located cells, large number of centrally located cells. We call them inner cell mass. For your easy understanding, I've colored it with blue color, right? Now the actual baby will be formed by these blue colored cells, that is inner cell mass. But the same is also surrounded by a green color ring-like structure called as trophoblast or the trophoectoderm. But this ring-like structure has no role in embryo formation. That is, this will not form anything in the baby, but yes, it will form two extra embryonic fetal membranes. That means two protective membranes around the developing baby. We call those membranes as chorion and amnion. I'm sure you remember amnion. In class 10th you have studied. So here what change has occurred, children? Few cells, they are in the center called as inner cell mass, which will be gradually changing into the embryo or the baby. But the trophoblast is surrounding them. No role in embryo formation, but yes, they will form two membranes called chorion and amnion. Now what happens further? Here we can say the structure will shift in such a manner that these centrally located cells will shift to one corner, right? How they will be shifted? See, at this stage, the structure is entering the uterus, and the uterine wall secretes a glycogen-rich fluid we call the uterine milk. And the milk, it enters the morula. That means the space that you can see between inner cell mass, the blue colored structures, and the green cells, that is the trophoblast, the uterine milk comes inside over here in this region. And from where it is coming? It is coming from the uterine wall, right? So this milk goes on entering inside the morula, and the pressure here is going to push the inner cell mass to one of the corners where it forms a knob-like structure. We call this as embryonal knob. Alright, so this embryonal knob only will form the baby. You can see the blastocele is the fluid-filled cavity now. How did the fluid come, children? From the wall of the uterus, and it entered the morula, right? So this blastocyst, this will be here, and this will be the 5th day after fertilization. So that means with the appearance of blastocele, a cavity here, morula changes into hollow blastocyst by the 5th day after fertilization. So all these changes from here, zygote up till here, take some 5 days, and we get a structure we call as blastocyst or blastula. Alright, so till here, just let me brief you up. The blastocyst formation, I told you, morula enters the uterine cavity. That means this entered the uterine cavity. The uterine milk was secreted by the glands in uterine wall, the glycogen rich fluid. Remember, glycogen is a storage food, product of glucose. Then uterine milk enters the morula and collects between trophoblast and the inner cell mass, causing formation of blastocyst cavity or a blastocele. So if we say what is the difference between morula and blastula, the clear difference is here it's a solid ball of cell mass, but here it's a fluid-filled cavity. We call it blastocele, right? Then the most important thing is the embryonal knob. When it is formed, this only will form the embryo or the baby. All right. And what is this trophoblast going to do? The trophoblast, also called as trophoectoderm, will not take part in the embryo formation, but this will form two extra embryonic, that means two membranes outside the embryo or the fetal membranes, and we call them chorion and amnion. So this is the first and second step only. I have discussed only cleavage or the segmentation and the blastocyst formation in these two pages. The changes are very, very simple, children. And let's quickly end up. Under cleavage, I explained repeated mitotic divisions in zygote, transforming a simple fertilized egg cell into hollow, spherical, and multicellular blastula. So you saw how the repeated divisions changed a structure like this, that is zygote, into hollow and multicellular fluid-filled blastula. Then, under cleavage, there were series of mitotic divisions. Interphase was short without growth. You can see here, the growth was held up, so there was no growth at all. Number of cells increased, but without growth, and this makes the blastomeres progressively smaller, and this is called compactation. You can imagine the volume is same, but the number of cells increasing, and this is called compactation. Then there was faster DNA synthesis, and finally increase in nuclear cytoplasmic ratio. So here, children, this is to be taken in some 2 to 3 videos. Today I am only explaining the first and second step under embryogenesis, that is cleavage and blastocyst formation up till here. 2 pages I am going to send you, and the next video I'll start from implantation. I'll request you in your textbook, this is given at page number 122 and 23. I have tried my best to explain you. Still, if there's any doubt, children, you can certainly ask any number of times. That's all for just now. Thank you.